everyone, we from Group 5 will show you a process of lifting the durability of Sambiloto herbal extract on bacteria Staphylococcus epidermidis. First, we'll make the concentration extract. In the researchers first made the concentration of ethanol extract of the bitter herba used, namely 20, 40, 60, 80% in DMSO solvent. To vary concentration of the solution, dilute the ethanol extract of the bitter herba with the solvent dimethyl sulfoxide or DMSO. At a concentration of 80%, 0.8 grams of herbal ethanol extract was which Sambiloto diluted with DMSO little by little on the watch glass until it can be put into the ependrove, then add 1 ml of DMSO to the ependrove. At 60% concentration, which 0.6 grams of ethanol extract of the bitter herba diluted with DMSO little by little on a watch glass until it can be put into the ependrove. Then add 1 ml of DMSO to the ependrove. At a concentration of 40%, take 0.5 ml of a concentration of 80% using a micropipette and put it into an ependrove and add 1 ml of DMSO, then homogenize. Last, 20% concentration, 0.5 ml of 40% concentration was taken using a micropipette. Put into an ependrove and add with 1 ml of DMSO, then homogenize. Here's the concentration of extract of 20, 40, 60, and 80%. Next, bacterial suspense. First, preparation of standard bacterial suspense solution is carried out by take 1 dose of rejuvenated bacteria and then suspend it inside NaCl 0.9% is sterile until the level of turbidity is the same as the McFarland standard 0.5. The turbidity is seen on a black paper background. If it is not cloudy enough, add bacterial colonies, and if it is too cloudy, then add 0.9% NaCl media. Next, testing containment zones. First, the test used the �𝘵𝘚𝘱𝘰𝘤𝘰𝘤𝘤𝘰𝘤𝘰𝘤𝘰𝘤𝘰𝘤𝘰𝘤𝘰𝘤𝘰𝘤𝘰𝘤𝘰𝘤𝑀 method without impregnation. Inoculate the bacterial suspension on Na media, evenly surface of the Na plate media using aseptic cotton swab. Incubation at 37 derajat Celsius for 15 minutes. Place a blank dish, moisten it with DMSO as a negative control and a 30 microgram chloramphenicol antibiotic dish as a positive control. Then soak the blank dish in the bitter herba extract at any concentration of 20, 40, 60, 80% until everything is wet. Attach a blank dish that has been soaked in each concentration and a dish containing 30 microgram of chloramphenicol antibiotic using sterile tweezers on Na media aseptically. The distance between one paper dish and another is arranged to calculate the inhibition zone. Incubation for 24 hours at a temperature of 37 derajat Celsius. Then after 24 hours, observations are made for the presence of an inhibition zone or clear zone around the paper dish and measurement of the radical inhibition zone of the bitter herba extract against Staphylococcus epidermidis bacteria. A total of 0.1 ml of the diluted bacterial suspension was sprayed on the prepared sterile fertilizing medium. The suspension in the cup flattened with a cotton swab sterile so that the colonies grow evenly on the media plate. Place in a 37 derajat Celsius incubator for 1 to 3 days. Stick the discs according to the line division that has been made in the middle. Pay attention to the discs to stick and not fall off. To another replication with the same thing, the petri dish is then wrapped, labeled group's name, the date of treatment, and the treatment group. Incubation at the temperature of 37 derajat Celsius during 24 hours. Afterward, look at the crystalline zones and a format and measured and used for source.
3 replications were carried out, where the �𝘵𝘢𝘱𝘩 method used 6 different types of sample treatment, including a negative control containing DMSO, a positive control, namely the antibiotic chloramphenicol, and 4 types of concentration variations of 80%, 60%, 40%, 20% bitter extract Andrographis paniculata. Nice. In the first replication the positive control obtained 19.05 mm, in the second replication the positive control obtained 19.35 mm, and in the third replication 19.475 mm with an average of 19.29 mm, using chloramphenicol. Chloramphenicol is a strong inhibitor of microbial protein synthesis. In practice, it was shown that the inhibitory activity of chloramphenicol against Staphylococcus epidermidis bacteria had an average of 19.29 mm. The negative control using DMSO did not have the ability to inhibit bacteria. In the first replication, the 80% concentration obtained 5.05 mm, the 60% concentration obtained 3.4 mm, the 40% concentration obtained 0.175 mm, and the 20% concentration obtained 0 mm. In the second replication, the 80% concentration obtained 2.15 mm, the 60% concentration obtained 0 mm, the 40% concentration obtained 0.275 mm,